How do methyltransferases affect our genes? Imagine your genes as a vast library of instructions. Methyltransferases are like tiny bookmarks or sticky notes that tell your cells which pages to read or ignore. These enzymes add small chemical groups called methyl groups to DNA or proteins around DNA. This process is a key part of epigenetics, which controls how genes are turned on or off without changing the actual genetic code. In DNA, methyltransferases mainly target specific sites called CpG sites, where a cytosine base is followed by a guanine base. When methyl groups attach to these sites, they usually turn off gene activity. This happens because methylation blocks proteins that activate genes from binding or causes the DNA to pack tightly, making it harder for the cell to read the genes. There are three main DNA methyltransferases. One called DNMT1 keeps existing methylation patterns intact when cells divide, helping to preserve gene regulation over time. The other two, DNMT3A and DNMT3B, are responsible for adding new methyl groups during early development or when the environment signals the body to change gene activity. Methyltransferases also modify histone proteins, which are like spools around which DNA is wrapped. These modifications can tighten or loosen the DNA, influencing whether genes are accessible for reading. Depending on where and how these histones are methylated, gene activity can either increase or decrease. These enzymes help maintain stable gene regulation across generations of cells. They also influence traits passed down in families. When methyltransferase activity is disrupted, abnormal methylation patterns can develop, leading to diseases like cancer or developmental disorders. Understanding how methyltransferases work is especially important for hereditary health. Methylation patterns can be inherited or affected by environmental factors, changing disease risk within families. Sometimes, even with the same DNA sequence, different family members show different symptoms because of these epigenetic changes. Testing for abnormal methylation can give doctors a clearer picture of hereditary disease risks. Researchers are also developing treatments that target methyltransferases to correct improper gene regulation in genetic disorders. In short, methyltransferases influence our genes by adding chemical tags that control gene activity. These modifications are vital for normal growth and health. They also play a big role in hereditary conditions, making them a key focus for understanding and managing complex genetic disorders. Knowing how these enzymes work helps us better interpret family health information and explore new ways to treat genetic diseases.